This empty neighborhood isn't from a lockdown. It's from a future where a simple infection is a death sentence. This is the silent world of antimicrobial resistance.
Awareness about antimicrobial resistance. According to WHO, AMR causes massive death, 1.27 million directly in 2019, and severe economic damage, with projected annual GDP losses of $1 to 3.4 trillion by 2030, and huge additional healthcare costs. Antimicrobial resistance, AMR, occurs wherever antimicrobials are used, in human and animal bodies, hospitals, farms, and the wider environment. A prime example is Staphylococcus aureus, which has evolved into MRSA, methicillin-resistant S. aureus, resisting first-line antibiotics. Similarly, VRE, vancomycin-resistant enterococci, and CRE, carbapenem-resistant enterobacteriaceae, are formidable bacterial threats in healthcare. AMR is not limited to bacteria, and fungi like Candida auris have developed resistance to multiple antifungal drugs, causing severe, untreatable infections. These pathogens demonstrate how AMR, driven by antimicrobial misuse, affects a wide spectrum of organisms, creating global health crises. For bacteria and fungi, antimicrobial resistance, AMR, is a critical survival mechanism. When exposed to a drug, susceptible microbes are killed. However, organisms that possess or acquire genetic resistance traits, like the ability to produce enzymes that destroy drugs, like ESBLs, or pump them out, survive. This resistance is not created by the drug, but is selected for by it. The surviving resistant microbes then multiply, passing their resistance genes to offspring and even to other bacteria through horizontal gene transfer. This allows the resistance trait to spread rapidly through a population. For the individual organism, AMR means it evades destruction. For the species, it ensures persistence and dominance in an environment saturated with antimicrobials, turning hospitals and communities into breeding grounds for them. Antimicrobial resistance, AMR, in bacteria occurs through 4 key genetic mechanisms. First, enzymatic inactivation, where enzymes like beta-lactamases degrade the drug. Second, target modification, where mutations alter the antibiotic's binding site. Third, efflux pumps, actively export the drug from the cell. Fourth, reduced permeability, limits the antibiotic's entry. These traits are passed vertically to offspring and spread horizontally by plasmids through conjugation. Fungi use analogous strategies. Target alteration via mutations in genes like those for ergosterol synthesis. Efflux pump upregulation to export antifungals and biofilm formation, creating a protective extracellular barrier that reduces drug penetration. In both kingdoms, antimicrobial use selectively eliminates susceptible microbes, allowing pre-existing resistant mutants to proliferate and dominate. This complexity allows a single pathogen to deploy multiple resistance mechanisms simultaneously, leading to multi-drug resistance and making AMR a highly adaptable threat. To fight AMR, a global One Health strategy is essential. The cornerstone is antibiotic stewardship, using antibiotics correctly, only when prescribed for bacterial infections, at the right dose, and for the full duration. This minimizes selective pressure. Other critical steps include preventing infections through vaccination and hygiene, surveillance to track resistance, investing in research for new drugs and diagnostics, regulating agricultural use, and public education on AMR dangers. These coordinated actions across human, animal, and environmental health are vital to preserve antimicrobial effectiveness for future generations.